will start with a new topic in this course now, which will be 3D NMR spectroscopy. This is something which was not covered in the previous course, and therefore this is completely a new topic. I would like you to pay a very careful attention to this set of slides or this topic, because now onwards we will be moving on to essentially all the protein NMR stuff, where we look at how 3D NMR is used for protein assignments and how 3D NMR spectroscopy is performed. So I would really recommend you to pay careful attention to each and every slide. So we will go through this very slowly and build up our knowledge. So let us start from very basics of 3D NMR spectroscopy. So the question first is, why do we need 3D NMR? So this example, this is shown here. In the case of methanol or ethanol molecules, small molecules, the number of peaks are very few. As you can see here, we know for ethanol it just 3 peaks, CH3, CH2 and OH. But when it comes to more complicated and large molecules such as proteins, the spectra 1D NMR spectrum becomes really complex. So you can see here, this is a schematic, or this is a representation of a protein structure, 3-dimensional protein structure, and each and every white grey color sphere here corresponds to a proton. So this is a ball and stick model of some protein. Now if I take NMR spectrum of a protein like this, each and every proton will give me a peak in this spectrum, because each and every proton in this molecule has a unique chemical environment, and therefore it has unique chemical shift value. So therefore they all will come in this region, but since all of them are basically the same type of protons like methyl, methylene or methine or aromatic or amide, their region chemical shifts range is all the same, is all of them come between around 0 to 11 ppm. So you can see, or minus 1 to 11 ppm. So you can see that in this range of 11 or 12 ppm values, all the proton chemical shifts come, and therefore if there are about 1000 atoms or more in the case of proteins, 1000 hydrogens, you will end up with more than 1000 peaks here in the spectrum, and that is something not easy to resolve, because 1000 peaks here compared to 3, definitely the overlap of peaks will be much higher in a 1D spectrum of a protein than a 1D spectrum of a small molecule. So therefore 1D NMR spectroscopy is virtually useless to work with for protein NMR, except that it gives us definitely some basic information of the protein. For example, when we record a spectrum of a protein like we did in the last part, we showed for 2D HSQC how it helps us to find whether a particular protein is folded or not folded. In the case of 1D, that information is also somewhat available to us. The chemical shift is spread well in the negative ppm, or here we can infer that this protein is well folded. If the protein were unfolded or denatured, there would not be such a spread of chemical shift as shown here. You will see more of lumps of peaks, they are all coming in different groups. So they will not be spread out or what is called dispersed as in a folded protein. But for any other purpose, 1D NMR is no longer useful. So therefore we need to go to 2D NMR, 3D, and in fact we need to go to even 4D NMR spectroscopy. So this is a picture showing schematically how and why and when we need the spectra, higher dimension NMR spectrum for structural studies. So you can see, if we are working with a smaller size protein, so typically in NMR we can divide qualitatively a protein into 3 different categories. One is called a small protein, which is somewhere less than 10 kilodaltons. So remember, in protein, when I say 1 amino acid, we will see this repeat again several times in the course, is about 110 daltons. So therefore if I have 100 amino acids, it will be approximately 11 kilodaltons. Okay. So any protein which is roughly a protein which is less than about 100 amino acid, I can call it as a small protein. Okay. If the protein molecular weight is somewhere between 10 kilodalton, that is 100 amino acid, so they can say here 10 kd, between 10 to 20 amino 20 kD, I can say it is a medium size protein for NMR from NMR perspective. And if the size becomes 20 to 30 or 40 kDa, we can call it as a large protein. In fact, greater than 40 kilodalton is very rarely in NMR looked at, and that is really large systems. So, in NMR we categorize proteins into different categories, and based on this particular category we need to adopt different strategies for looking at the structure of that protein. So, typically when you have a small protein or a peptide, we do not need 3D NMR, 2D NMR is sufficient. Therefore, when in the beginning stages of NMR, before isotope labeling was invented, all the proteins which were solved were about 50, 60, 70 amino acids, and they were all less done by 2D homonuclear NMR, which we have covered in the previous part. When you go to slightly bigger, like below 10 kilodaltons as shown here, or somewhere from 8 or even 8 or 6 kilodalton onwards, you can start doing 3D NMR. You can start one has to use 3D NMR for structural studies, because of the resolution problem which we will deal with or discuss again, and that is comes in this particular range. But when you go to larger size proteins, that is 20 to 30, which is roughly sketched here, not really in that range, but it is shown in this from 20 to 40. Here you need to go beyond 3D NMR. So, you need to do a higher dimensional 4D NMR, which we will not do in this course, because that is a little complicated when it comes to analysis, but the basic idea of 4D comes from 3D, and basic idea of 3D in fact comes from 2D. So, we will cover up to 3D NMR in this course, but as you go higher and higher, as you can see here in molecular weight, when you go to very large systems, you have to actually do 4D NMR with more special techniques called deuteration, TROSY, etc. Deuteration basically means replacing every hydrogen in your protein, every exchangeable non-exchangeable hydrogen with a deuteron or deuterium. Okay, so that is a deuteration that is done with the molecular biology approaches, as we will see in the case of isotope labeling, and there are special NMR experiments which were actually invented to tackle the challenges of large proteins, and that is called as TROSY, and combined with 4D NMR you can actually attempt to look at structures of protein in very large molecular weight, which is around 60-70 kilodaltons. But typically 90% of the systems we normally study by NMR falls in this range, and most of them actually we use 3D NMR, which falls in this 20, 25, 30 kD, and we will look at basically how 3D NMR can be used for protein structure and assignment. So, we basically again coming to this question, why do we need 3D NMR? So, there are three basic things which we will touch upon. First is to see that how 3D helps to resolve peaks that are actually overlapped in 2D. So, we saw that actually 2D itself helps us to resolve peaks which are overlapped in 1D, and we saw some examples of that. But sometimes even in a 2D, a peak can be overlapped with another peak. So, two or more peaks can overlap with each other in a 2D spectrum. So, in such cases 3D will help us to resolve. We will see also how 3D helps to obtain new chemical shift correlations. Again, this is similar to 2D. In 2D we saw we got new correlations, chemical shifts, and that will be also extended to 3D, and again we will see how new correlations can be obtained. And third thing which we will see with 3D NMR is when do we actually need it, when do we use it, at what stage of our study of structures of proteins would we need 3D NMR experiments. So, let us start from the first point here, that how does 3D help to resolve peaks which are overlapped in 2D spectrum. So, this is I am going to give a very qualitative example now. This is a kind of a hand waving example, but this is just to illustrate the point. So, I will give a non-real example, then we will go to more cases from a real system. So, let us say you have a directory with you and you are trying to identify people from a directory. So, you are basically given, you know the name of a person, let us say only the name, and you try to search in the directory. So, in this case the name I am going to take example of my name, that is Hanudatta Atreya. So, let us say you have the first name Hanudatta with you, then you look at H, but then let us say you end up finding 2 Hanudatta names. So, 2 persons having the same first name, then what do you do in such cases? How do you resolve this degeneracy? So, this is called a degeneracy because both are same first name. So, obviously, you have to look for a surname if you want to distinguish this person from this person. So, if you use a surname and a first name, then actually you are going into a 2D mode. So, in a 2D you are using one dimension as surname, other dimension as first name, and then you will trying to figure out whether this Hanudatta has a different surname compared to this Hanudatta. So, if they do have a different surname, then they are resolved, means you have separated them and you can say there are 2 different people. But suppose the names are also having the surname is also same, that means the 2 persons having the same first name and surname, then how do you resolve this problem? So, this is you see in the 2D, that means using surname as a independent dimension and a name as another dimension or a variable, we were not able to resolve 2 people who have the same first name and the surname. So, obviously, we have to go beyond 2D now, and suppose I use another dimension, I add one more dimension here and put the dimension as address of the person, because the directory normally has a name, surname, address, phone number, etc. So, I expect that okay, maybe the surname of these 2 guys will not be the same, sorry, the address will not be the same. So, if the name and surname are same, maybe the address will separate them or differentiate them. So, if I use a 3D approach where I look at now address as a criteria for separation, you can see here that this person may live in North Bangalore, whereas another person may be in South Bangalore. So, I have separated them by using address as another dimension. So, you can see from this example what essentially we are doing here is we are adding independent dimensions to resolve the peaks. So, you can think of these as peaks, you are trying to separate peaks which have the same chemical shift. So, if you think of this as a chemical shift now, first name as a chemical shift, proton chemical shift, this H had this peak at this had the same proton chemical shift. So, instead of 1D, I then added a carbon chemical shift to it, directly attached carbon. So, that for example, becomes an HSQC. There again probably the both proton and carbon are same, that is both name, surname in this case are same. So, again there was an overlap in a 2D. So, I added one more dimension or one more chemical shift, let us say nitrogen, and that molecule, if it has nitrogen, of course it has to have nitrogen, otherwise we can't add a dimension. But let us say those 2 molecules had different nitrogen chemical shifts, then they can be resolved. So, we are essentially relying or connecting more and more chemical shifts, so that peaks can be resolved. So, this is one of the aims of 3D NMR expert spectroscopy, is to resolve what could not be resolved in a 2D spectrum. So, let us now look at actual molecules. So, let us say we have these 2 molecules, serine and cysteine. So, serine and cysteine are actually 2 amino acids in a protein. So, many proteins have both of this, this is also rare although in molecules proteins, but let us say we have this both present in a molecules. So, if you look at the structure, they are nearly the same, they look the same, except there is a sulfhydryl group here and there is a hydroxyl group here. So, there is a slight difference in that sense, but if you look at the chemical shifts of A, this alpha hydrogen here and here are almost the same within the deviation expected from molecule to molecule or structure to structure. B, that is this proton B1, B2 here, these 2 methylene protons and these 2 methylene protons are slightly different. Okay. So, if I record a 2D toxi spectrum of a mixture of serine and cysteine, so I have mixed these 2, they can be the same concentration or different. So, for example, in a protein I may have 3 or 4 cysteines, I may have 2 or 3 serines. So, they are actually like a mixture now. So, if I mix the 2 and add, they come very close to each other, you can see here, although there is some over differences because of this chemical shift values, but the 2D toxi spectrum is able to slightly differentiate, but still there is some overlap. Okay. So, now the question is, can I use a 3D NMR experiment to separate or further resolve or make it better separated for these two molecules. So, I can do that with the 3D. So, you can see a 3D, how do I do that? I need one more chemical shift. So, this is something I mentioned in the previous slide, that we had name and surname were same, but we use a third possibility, that is address to resolve. Similarly, here I can go to, I have these three which are almost similar. Okay. I do, although these are two are different, but let us assume these are all very similar, but these, look at this C beta chemical shift of these two amino acids. There is a huge difference. Okay. So, this is slightly a mistake here. This should be actually 63 ppm and this is 32 for serine. So, you can see here that a cysteine comes around 32 ppm, that is it is when it is SH in SH state, and serine beta 1, that is the protons at the carbon for beta, that is this carbon. Now, remember we are looking at this C beta here, comes around 63 ppm. So, you see there is a very big difference, 30 ppm difference between the two, but all other values seem to be similar. So, therefore, if I use C beta as a discriminator or a third dimension, means I have two protons in a toxi, but I can add one carbon dimension and make it a 3D HSQC toxi, then in such a spectrum I should be able to completely separate this spin system, this family from this family, right. So, that is something what we can be done using a 3D HSQC toxi, which is done by combining a 2D HSQC with a 2D toxi. So, like this, by combining two dimensional NMR, I can construct or I can create a single 3-dimensional NMR spectrum experiment, wherein now I am using carbon as a third dimension. So, if you remember this plane here, this what is shown here, this green peaks and red peaks, they were actually the toxi planes. So, if I merge these two, if I join these two, it becomes the 2D toxi of the mixture. But now I have separated them by using C beta as a separator or a discriminator or a third dimension, and by virtue of that I am able to separate the cysteine family of spin system from the serine family. So, this is one of the examples how we are able to separate two molecules or two amino acids just because we had a very good possibility in a carbon dimension to distinguish them. So, like that we can construct a 3D experiment. So, not only serine and cysteine, it would be possible to also separate some other two amino acids which are having close chemical shift in protons, but which have very good C beta or C alpha or C gamma, some carbon differences, and in HSQC toxi we should be able to separate them. So, this is an example of how a resolution can be obtained. Now, let us come to the protein NMR experiment which we normally use, which is called the 15 HSQC. We have looked at this in detail in the previous classes. Now, we will again look at, say, let us say in a 2D, as I mentioned, each peak here corresponds to one amino acid, and that is this shown here. So, this is the peptide bond. So, we have each amino acid has an amide except the N terminus and proline, but this amide proton and amide nitrogen come in as a peak. But suppose I have a case like in a protein, we have a very complicated structure. It is always possible that two amino acids have accidentally the same chemical shift value, that is they have the same amide proton and amide nitrogen chemical shift. That means, this amide proton and nitrogen for both of them are same. So, therefore, these two peaks are now sitting on top of each other. So, normally we will not be able to distinguish these two peaks because they are exactly or very close to each other. So, they will come as a single peak, appear as a single peak. I have shown it different here, different color just to illustrate, but in a real spectrum you will not be able to separate the two, they will be overlapped. So, in such cases how do I know that there are two peaks here? I will not be able to figure out looking at the peak, because the peak will look like a single peak. So, I have to do something more to achieve that separation or resolution, and say that yes, there are 2 peaks here, not 1. So, for that I need to go to 3D NMR now. Now, what do we do here? What kind of chemical shifts? Another dimension we need here, right, like we showed in the previous slide. So, we need to add a third dimension, another chemical shift. So, there are varieties of possible options. I can use C alpha as a discriminator. That means it may be possible that the C alpha chemical shift of these 2 amino acids are not the same. This has a different C alpha and it could be having a different. This is a guess. I do not know, it may be or may not be. Or I could use CO as a discriminator. You see, this has a CO beside it, beside this N, and there is a CO beside this N, and every NH pair in the amino acid in the protein will have a CO of the previous amino acid. So, I can use CO as a discriminator, and it is probably possible that amino acid X has a different CO compared to amino acid Y as a in chemical shift terms. That means, the CO chemical shift of these 2 are not the same. So, that is a possibility. I can use that as a third dimension. So, that these are different experiments. For example, if I use CO to distinguish the 2 amino acids, that means I am adding CO dimension, I am going to add CO dimension to this spectrum. So, it will become HNCO, because CO will be another dimension. So, it becomes a 3D experiment wherein I have 3 chemical shifts H, N and CO correlated with each other. Okay. So, let us see how that can be done. So, this is exactly what is shown here, that they have 2 amino acids somewhere in the sequence, and accidentally, remember, they are just by chance they had the same chemical shift values. So, therefore, we need to separate them or distinguish them to know that there are 2 peaks. So, what I can do, I can probably use CO, which is close to the nitrogen. You see, there are close to it. So, every amino acid has a CO attached to nitrogen, and that can be used because they may be different. That means, CO may be different for this amino acid from this amino acid. So, that is again an assumption, we do not know that will be true or not, but we can take a guess and then design a 3D experiment HNCO, which will now help us to resolve those 2 peaks. So, such an experiment will look like this. So, that is a spectrum of the 2D spectrum which I showed you with an overlap here. Now, if I record a 3D, now 3D will look like this, because it has 3 dimensions H, N and CO, and the CO is now acting like a separator, like a discriminator. So, you can see each of the peaks of here shown here will come in different planes, because their CO values may not be the same. Okay. So, if we are able to separate those peaks, for example, these 2 peaks here which were actually overlapped in the 3D spectrum or in the 2D spectrum, we are able to separate them because of this nature of CO, which helped us to distinguish the peaks. So, this is an experiment, we can name it as HNCO, because we are correlating H, N and CO. So, this is the simplest 3D experiment you can think of. In fact, this is the most basic 3D NMR experiment known and very sensitive in nature. But one thing you have to keep in mind is that we have nitrogen, which is N15, and carbon also has to be labeled C13. So, basically we need to label, isotopically make this molecule enriched or rich with C13 and N15, then only we can perform this experiment, because carbon is natural abundance C12 is only 1% and natural abundance of nitrogen is 0.3%. So, if I want to do an experiment like this on an unlabeled, means natural abundance on any molecule, it will be virtually impossible, because I will have very low abundance of this isotope which are required for NMR. So, therefore, I need to label the molecule or protein with C13 and N15, and therefore, all these 3D experiments which you will be seeing henceforth are actually carried out on labeled protein samples. So, that is something which we will come across, we will see that as we go on, because without labeling it is not possible to do this experiment. Now, let us see what are the different things in 3D NMR one should understand. So, as I showed you in the previous slide, a 3D experiment but let us look at the different projections. Now, this is something which one has to understand very carefully. So, let us say this is taken from Google. You can see, suppose you have some drawing or some 3-dimensional object like this, and suppose I want a view, different views of this from different sides. So, typically in engineering we talk about front view, side view and top view. So, that is shown on this side here. Your top view may look like this, front view if you are looking from here it may look like this, side view may look like this. So, similar to this kind of picture we can actually have also for NMR spectrum, 3D NMR spectrum. So, this is the 3D spectra which I showed in the previous slide. So, you can see this is 3 dimensions. Now, if I want to look at different projections of this similar to that, how will they look? So, let us start from the very basic front view. So, if I am looking from this angle, basically what will happen is this CO dimension will disappear like here. When you are looking from the front side, you do not look, the side dimension disappears. Okay. So, like that you will get with only HSQC spectrum back. So, actually in a 3D, the HSQC spectrum is one of the projection of a 3D, and that is something which we should know, because we took an HSQC and we use carbonyl or CO as a discriminator, as a separator. So, therefore, if I did not have this dimension, I would have got a standard HSQC, and that is what is one of the projections of a 3D spectrum. Okay. So, this has this overlap which we could resolve, but if we do not have this dimension it does not get resolved. Now, let us see how the top view will look like. Top view basically means you are looking from the top here, similar to this kind of a view perspective, and here the peaks are now coming in different position, they are no longer same as this. Why? Because if you look from the top, this now we are looking at the HCO axis. So, it is not nitrogen here, it is not HSQC, it is a proton carbonyl projection. There it depends on the carbonyl chemical shift where the peaks will come. The proton will be the same as this proton. For example, this one here, let us say has a proton peak here which corresponds to 6.5. So, let me write down to make it more clear. If this proton chemical shift is, let us say 8.5, this proton here also will be 8.5. Okay. So, that will not change because we are looking at the same proton axis in the both cases, but the carbonyl will depend on what is the carbonyl value, CO value in this dimension. So, therefore, the peaks looks now rearranged in a different order compared to this because of this CO dimension. Now, I can look from the side view. If I look from the side view, I will get CO nitrogen correlation. Okay. So, this is again a different, something completely different from these two, because now I am getting CO and N value. So, if you look at N now, this N will remain the same. So, for example, let us say this 3, if this had 120 ppm, this also will be 120. So, that will not change. So, this is just an example to show you that this chemical shift of 3, which is nitrogen part, is same as this nitrogen here, because this axis and this axis are the same. So, we can see in NMR 3D NMR experiments, we therefore have to focus on projections of NMR spectra to understand what is happening, and this is how NMR spectra 3D are analyzed. We will look at again a few more examples later. We will quickly now just look at a projection example for a 3D HSQC TOC. So, this is a 3D HSQC TOC which I showed you just a few slides ago, how that helped us to resolve cysteine and serine, but it will have a peak pattern like this. So, if you look at this box, 3D box, if I look at different view, the top view will look like this, because I am looking from the top, from here. So, the top view is basically coming from here. This is the top view, and you can see that is basically a carbon proton plane which is shown here. Okay. And then if you look at front view, that is proton TOC is shown here, and then side view, when you look from this side here, you will see a proton regular proton TOC, and this is basically carbon proton TOC. So, I am looking from this angle. So, you can see this blue plane here, this blue plane is basically this blue plane here, okay, proton carbon HSQC toxi. So, this 3D HSQC toxi, when look at the different projections, they are actually 3 different experiments, but they all got combined into a single experiment and I got a 3D spectrum out of it. So, basically a 3D experiment, if you look at the different projections of the 3D, are actually 3 different spectra which normally would be collected with different pulse sequences, like this has a separate pulse sequence we saw in the previous part, this has a separate pulse sequence, this has a separate pulse sequence. We have seen actually the pulse sequences of all these 3, but these 3 are not separately collected and joined and made like this. What is done is this 3D HSQC toxi is a single experiment which is recorded in a single data set, but if you look at its projections, they will end up separating into 3 different spectra. So, we normally do not analyze projections so like this, but it is useful to know how a 2D projections of a 3D look like. So, we will continue this aspect in the next class, look at few more examples and then we will see how actually a 3D spectrum is analyzed and interpreted.